Good morning, my dear students. Today we are discussing about the gastrulation in chick. So the term gastrulation is applied to the process which produces the gastrula. Gaster, the word meaning that is stomach. It is a process of highly coordinated cell and tissue movement whereby the cells of the blastula are get rearranged to form three germ layers, that is ectoderm, mesoderm and endoderm. The numerous cells of blastula formed due to cleavage are given new positions and neighbors during gastrulation, resulting in the establishment of the multi-layered body plan of the adult organism. During gastrulation, the blastocelic cavity is obliterated and a new cavity called archenteron is formed. Gastrulation can be defined as the dynamic process during which the major pre-assumptive organ-forming areas of the blastula they get rearranged and reorganized in a way which permits their ready conversion into the body plan of a particular species. Thus, gastrulation it is nothing but embryo's way of laying down its body plan. So, gastrulation is a formative process by which the three germ layers, which are the precursors of many embryonic tissues, and the axial orientation are established in embryos. So, during gastrulation, bilaminar embryonic disc, here you can see in the picture, is converted into trilaminar embryonic disc. The embryo during this stage, what we call it as gastrula. So gastrula begins within 4 to 5 hours after the onset of incubation and it gets completed by about 22 hours. A gastrulation in chick embryo can be divided into two phases or two important events which happen to take place during gastrulation. That is endoderm formation and second one that is primitive streak formation and movement of cardomesodermal elements. So before going to the process of gastrulation, there are two types of morphogenetic movements, nothing but the cell movements. So that is emboli and epiboly. So epiboly, it involves only the epiblast. You know about what exactly epiblast, which contains the cells of ectoderm, mesoderm and notochordal cells. So this emboli includes convergence, invagination and involution. So the formation of primitive streak and the head process, it is only because of the process that is emboli. So second one that is epiboly. It includes the overgrowth of ectoderm or epiblast and also the hypoblast. So here in this picture you can see exactly the cell movements which happens to take place during gastrulation. First one that is invagination, that is infolding of shell sheet into embryo. Involution, that is turning in of cell sheet over the basal surface of an outer layer. So ingression, that means the migration of individual cells that get inside the embryo. Delamination, that is splitting or migration of one sheet into two sheets. So second thing that is epiboly, that means the expansion of one sheet over other cells. So this is what happen during the gastrulation, that is emboli and epiboli, nothing but the cell movements. We call it as morphogenetic movements that may take place in different kinds. So first we have already discussed two major events, that is first one, that is formation of endoderm. So endoderm of hypoblast, you can see here that will develops as a single layer of cells inside the blastocele. In this picture you can clearly make out which has been taken the section, and the hypoblast that will develops as a single layer of cell that will get inside the blastocele. After the formation of endoderm only, you can see the upper layer, what we call it as the epiblast. So this is where the formation of endoderm takes place during the gastrulation process. Second major event, that is formation of primitive streak. The second step in gastrulation, that what we call it as the formation of primitive streak. So go looking at the picture. So here various mesodermal and endodermal cells, that means which are present there, the cells forming notochord of the epiblast, they converge towards the posterior edge of the area pellucida. Two areas have been formed, that is outer one area opaca and inner, that is area pellucida. So you can see the conical thickening in the middle line, that what we call it as the initial primitive streak, where we can see the starting stage of gastrulation. So this will appear after 6 to 7 hours of incubation. When there is a number of cells, they start to migrate, the primitive streaks, they also grows anteriorly because of the proliferation of its own cell, as well as there will be addition of cells, and that will from anterior to the lateral parts of area pellucida. So in the third picture you can see that what we call it as definite streak stage, the elongated axis of primitive streaks which marks the anterior posterior axis for the future embryo. So here also it eventually extends up to 3/4, that means the 3/5 of the entire length of the area pellucida. So this fully developed, we call it as the definite primitive streak. See picture. And usually it get complete in 18 to 19 hours of incubation. So the area pellucida also become pear shaped along the middle of the primitive streak. When it is fully developed, there runs a narrow furrow, that what we call it as primitive groove. In the picture D, you can see different regions in the area pellucida. Starts with the primitive streak, and you can see the primitive groove there. At the anterior end of the primitive streak, there is a thickening, what we call it as primitive knot or Henson's node. So the center of the Henson's node is excavated to form a funnel-like depression, that what we call it as the primitive pit. So here you can see there is a primitive groove, and this primitive groove they get a fold, that is primitive fold, and there is a knot-like structure, that is primitive knot or Henson's node, and depression where you can see that we call it as the primitive pit. The movements in the blastoderm leading to the final placement of the cell in the hypoblast and to the formation of primitive streak in the epiblast, and this we can call it as pregastrular movements. So another two important, after when there is a formation of all these, you can see invagination and involution. At the stage of short primitive streak only, the cells of the blastoderm, that is the outermost layer, begin to migrate into the blastocele cavity, which is lying between epiblast and the hypoblast. Here you can see the presence of red color cells. You can see there, those are, we call it as migrating cells. So this immigrating cells, they are replaced by more epiblast cells. They converge towards the streak area. So the inward migrating cells, they also spread out the sideways, you can see here, and also forward from anterior end of the primitive streak. The notochordal cells, they also immigrate through the primitive pit. Endodermal cells invaginate through that part of streak which lies just behind that of the primitive pit. The primitive streak elongates along with this area pellucida, and also it gets elongated. As the primitive streak starts growing, the cells from this region will involute into a space between, that is, epiblast and hypoblast, and the process we call it as immigration. So these immigrated cells will become future precaudal plate notochord, and also they can become the mesoderm. So the mesoderm is formed are of two layers in front of the primitive streak. An area without mesoderm is present, and that what we call it as proamnion. So at this place later the head will get developed. After 48 hours of incubation, the proamnion is occupied by mesoderm. So later the mesoderm, it is divided into dorsal, intermediate and lateral mesoderms. So now the notochordal cells, they arrange themselves in a form of cylindrical rod called notochordal process. It will begin at Hansen's node, or what we call it as primitive node, and it will starts to grow slowly. Because of its growth, the primitive streak is slowly getting reduced. So at the end of the gastrulation, the primitive streak is reduced and it is incorporated into tail bud. The dorsal mesoderm is located on either side of the notochord, and that what we call it as the segments, and they are also called it as the somites. The first pair of somites will form 21 hours of incubation. Afterwards, for every 1 hour, one pair of somites they will get add to that. So the 24 hours old embryo that will contain 4 pairs of somites, the intermediate mesoderms that will connect to the dorsal mesoderm with lateral mesoderm as a stalk. Afterwards, it undergoes the segmentation, and that will give rise to kidney. So the lateral mesoderm extends on periphery of the embryo. You can see intermediate and all types of mesoderm there. This will develop periphery of the embryo, and it has been divisible into extraembryonic and embryonic mesoderm. The lateral mesoderm that gets splits into 2 layers. So in this picture you can clearly see that is the upper one we call it as somatic mesoderm, and the inner one we call it as splenic mesoderm. So ectoderm and somatic mesoderm that will form somatopleure, and splenic layer and the endoderm will form, that is the splanchnoplure. In between these 2 mesoderms, that is somatopleure and the splanchnoplure, the space what we call it as the coelom. So thus at the end of the gastrulation, specific organ forming areas they started to develop. So here is the cross section of an early chick embryo. You can see that is the first one, that is the formation of endoderm, and along with that you can see that is the second step, that is the formation of the primitive streak, and later it get degenerated and the mesoderm development, and there exactly it has been divided into dorsal mesoderm, lateral mesoderm and intermediate mesoderm, and latter mesoderm has been divided into somatic and splenic layer of mesoderm. So when you see, this is what we call it as the fully formed gastrula, where you can see all these important structures when it is fully formed. So gastrula is fully formed when the primitive streak that has been completely disappears. The fully formed gastrula contains three germ layers: ectoderm, chorda, mesoderm and endoderm. The ectoderm and chorda mesoderm remain in continuity along the axis of primitive streak. The endoderm is also united with the mesoderm and ectoderm at the anterior and the posterior end of the streak. So this is about the gastrulation process what you found in chick. Thank you.